Hello everyone, I'd like to thank the organisers for giving me this opportunity to present some work that we've been working on for a few years, which is to identify transmission blocking compounds, and thank you for Michael for that very nice introduction. So what I'm going to present today is some work that we've done in collaboration with Vicky Avery with the gracious support of the Bill and Melinda Gates Foundation, with Omar being our program officer, Omar Vandal. So a number of years ago, Sophia Jali in my lab, who was a graduate student, and I started visiting the issue of which antimalarial drugs would be effective at blocking transmission, and we noticed that the literature was quite varied in sort of the robustness of the assays being used to identify gometocytocidal compounds. There were reports sort of scattered throughout the literature with a diversity of assays, and so we decided that we would develop a luciferase, a transgenic reporter line that would allow us to sort of systematically move through the antimalarials and see which ones of those in current clinical practice actually are effective at blocking gametocyte development in vitro. So the way we did this was to develop luciferase-based reporter lines that we could use to assay inhibition of parasite development throughout these 5 stages of gametocyte maturation in vitro. So the genetic approach we used here took advantage of an earlier system we developed, which is this ATB-ATP based method of integrative recombination, where we introduce an ATB site into the genome in a stable integrated manner without a selectable marker, and then we can introduce a plasmid that's replicating episomally with this homologous recognition site we express in trans and integrase, which catalyzes this recombination event leading to the integration of this reporter locus into the genome. We did this in the NF54 parasite provided by Steve Hoffman and his colleagues that had recently been pathologized through humans. So these are highly infectious. They make good numbers of gametocytes in vitro. And this was sort of the basic schematic of the parasite that we have, expressing a fusion of GFP luciferase with the BSD blastocidin selectable marker upstream. And then we could basically introduce whatever promoter we wished upstream of GFP luciferase to confer stage specificity of expression. So these were 3 reporter lines that we worked with initially: PFS16, which peaks early in gametocyte development but continues to be quite well transcribed throughout the later stages; PFS48-45, which is more mid-stage, stage 3, and this MAL8P1.16, which is more highly transcribed later in gametocyte development. And so using these, we were able to screen the first-line antimalarial drugs for transmission for gametocytocidal activity in vitro. So the way we did this was essentially to mimic a 3-day course of treatment. So gametocytes were induced in vitro, and then drug was applied for 3 days. And so just to sort of benchmark the concentration, we determined the IC50 of our test compounds against asexual blood stage parasites, and then we chose either half X, 1X, or 5X, the asexual IC50, for these 3-day treatments, and then we used luciferase as the readout of inhibition of parasite growth. And the luciferase reaction is an ATP-dependent reaction, so as soon as the parasite dies and you start to see attrition of ATP levels in the cell, you quickly see this translate into an effective readout of inhibition of parasite growth, leading to parasite death in vitro. So we ran this through these different assays. We made measurements every day. So I'm giving the example here for methylene blue, where we treated either synchronized early stages, mid stages, or late stages, and shown here are the dose-dependent inhibition curves for the half X, 1X, and 5X, asexual stage IC50, and you could sort of essentially establish a concentration response curve across time here. So this is not an IC50 so much as just looking at the slopes of inhibition at these different concentrations. We can then take these data and make a cumulative analysis of the ability of the different first-line antimalarial drugs as a function of the different stages, looking at the reduction in gametocyte development here on a log 10 scale. And so what we see here, for example, with atovaquone, many of the current licensed first-line antimalarial drugs are quite potent against stages 1, 2. So you can see this initial drop essentially in gametocyte development in the early stages. But for a number of these drugs, for example, atovaquone, tofenaquine in vitro, this is not the metabolized form, you could see it basically plateau out as these drugs lose their ability to inhibit the late-stage gametocytes. The one that really stood out quite clearly from all the others was a compound named methylene blue, which is a very old antimalarial compound. It was actually the first compound reported in a clinical trial to be effective against malaria. This dates back to the late 19th century with Ehrlich. This is a chemical dye, basically from the German synthetic dye industry. A very effective compound, and what was quite remarkable was very effective reduction in gametoside development, including on the late stages in vitro. We then set up a collaboration with Steve Hoffman, a long-term collaborator, heading Scenaria, to look at the ability of these compounds to inhibit transmission of late-stage gametosides to mosquitoes as measured by dissecting the midguts of these mosquitoes and then scoring them either for presence or absence of infection or for reduction in the oocyst numbers. And here we're looking at the block in transmission, implying the percentage of mosquitoes that are sterilized, that have no oocysts, following treatment for 3 days of stage 5 gametosides prior to feeding these gametosides through a membrane feed to the mosquitoes. And you can see methylene blue here at either quarter X or 1.25X the IC50 against asexuals was extremely effective, 100% or close to 100%, in essentially sterilizing these mosquitoes. And these are very low concentrations. I should point out that methylene blue against asexual parasites is in the order of about 20 to 25 nanomolar. So at about 30 nanomolar here, we were basically getting sterilization of mosquito infection. Now, this comes with caveats. It's known that methylene blue has certain side effects. It's a synthetic dye. You can't remove that dye component. One of the side effects is it turns your urine blue, so it seems to not be necessarily so popular, but I do think that it's an interesting mode of action. It's definitely an interesting pathway because of its potency here against these late stages. I should also point out, we've done a lot of work in collaboration with MMV on raising resistance to antimalarial compounds in the preclinical development pipeline, and methylene blue really does stand out as one of the compounds for which it's essentially been impossible to date to get stable resistance in vitro. So there's something about its mode of action. We think it's a redox perturbing agent that makes it refractory to facile acquisition of resistance by falciparum. So once we had these the assay and we could show that we could robustly identify gametoside-acidal compounds, we submitted a proposal to Bill Melinda Gates Foundation, which was funded to allow us to initiate a collaboration with Vicky Avery and her group, her high-throughput screening group at Griffith University, showing here to turn this into a high-throughput screen to look for inhibitors of either early or late-stage gametosides. So this is the essential approach in vitro. We start with parasites that we synchronize on day -6. On day -3, we then put these over a magnet to purify these late-stage parasites. We drop the hematocrit. We allow them to go to hyperostemia with nutritional stress to trigger the induction of the sexual stages, and then we magnet purify to eliminate any remaining schizons, and we treat it with NAG, N-acetylglucosamine, to eliminate any remaining asexual stage parasites so that we can begin the assays with purified gametosides. So this is a screen that Leonardo Lucantoni and Vicky's lab set up. We're using luciferase then. He's able to plate these. He goes up to 384 well plates. They've tested about 30,000 compounds so far, including compounds from the MMV malaria box, from the GNF Novartis malaria box. These are asexual blood stage active compounds. Some preliminary work on a St. Jude screen and some recent screens as well. So this is just to show also the Z prime on these is perfectly adequate. It's about 0.8, so this is proving to be a robust screen. But I won't go into the details of the screen itself. These are some of the data on the MMV malaria box and the Novartis GNF malaria box, plotting basically the activity against early stage gametosides versus asexual blood stage parasites for a subset of compounds here. And what you can basically see is that on average we see about a 3-fold or to 4-fold higher IC50 against gametosides versus asexual blood stages. For the malaria box, it's about the same here with the Novartis GNF. We have a number of sub-micromolar inhibitors, but there are a few important points I want to emphasize here, one being that immature gametosides tend to be less sensitive than asexual blood stage parasites to most antimalarials. We cannot a priori assume that asexual blood stage inhibitors act on young gametosides, and I think that's an important point. Many times people have assumed that if it's active against an asexual blood stage parasite, it will be active against an early gametoside, and that's mostly, I think, a consequence of the early emphasis of antimalarial drug use on inhibitors of basically the DHFR inhibitors, the antifolates, as well as the heme-interacting drugs that I think biased our opinion of this correspondence between action against immature gametocytes and against asexual blood stage parasites. And I think that if we want to have a large library of a large collection of hits against immature gametocytes, we need a high-throughput screen on a chemically diverse panel, not necessarily ones that initially show activity against asexual blood stages. For the late stage, this has come along a little bit later. We use the PFS16 as a reporter because we still get a good signal on late stages. Pietro Alano will show you some work later where we've collaborated with this group to help identify more potent reporters that have a higher signal in late stages. But for now, Vicky's group has been using the PFS16, and we see here about 10,000 compounds screened so far with some recent libraries that she's included in this. And I want to focus the last part of the talk on some compounds that we have found that appear particularly promising. Here are 4 compounds that came out that had the property of being highly active in vitro against asexual blood stages, as well as early stage gametocytes and late stage gametocytes. These data came from a luciferase-based screen. This was from another screen in Vicky's lab using GFP to identify activity against late stages. But you can basically see promising activity here with low nanomolar double digit against early stages and still quite respectable activity against late stages. Now, of these 4 compounds, I should point out that the 5668, the 5660, and the 5640 belong to one chemical class. They're called the dihydropyridones. This is a muscarinic acid-like compound. And so we took these compounds, and these come from the GNF malaria box of about 3,800 compounds with asexual blood stage activity. In this case, for this class of dihydropyridones, we're looking at in the range of 5 to 18 nanomolar against asexual blood stages. So really quite potent and attractive properties against these 3 stages. So we've gone ahead and worked with Vicky's group and also with Michael Delves and Bob Sinden and also Steve Hoffman to look at the activity against mature gametocytes. These are the data with exflagellation at 3x the asexual IC50, just to sort of standardize the data here. Essentially, methylene blue effectively completely inhibited exflagellation at very low concentrations. Here are 2 of the compounds from the dihydropyridone series, and you can basically see very good knockdown in rates of exflagellation at these low sort of nanomolar concentrations. We also worked with Steve Hoffman to look at inhibition of transmission. Again, this is the mean number of oocysts per midgut. You can see with the untreated, we're looking on average, these are 2 different experiments, around 15 oocysts per Anopheles midgut in these experiments. With DMSO, it was somewhat similar. The second experiment, it climbed up a little bit, but that didn't really have any negative impact. Methylene blue, as we knew before, essentially sterilizing the infection here. And here are data with these 2 dihydropyridones showing substantial knockdowns at the 3x values, so still around the 50 to 60 nanomolar concentration in terms of reduction of oocysts in the midgut of these drug-treated parasites in the mosquito. And again, showing here the percent transmission blocking then with the 5668, 5660, getting close to 100% reduction in transmission blocking with these compounds. So we've been interested in the mode of action of these compounds. Do they have a novel target? So we went ahead in the lab and selected for resistance to these using protocols similar to those we use, essentially the same as those we use to select for resistance to compounds in the MMV preclinical pipeline. And we then subjected these to whole genome sequencing of isolated clones from these resistant parasite cultures. The sequencing was performed by our close collaborator Elizabeth Winsler and Victoria Dorosky in her lab, as well as Philip Heinrich in my lab. Both ran whole genome sequence analysis to identify the genes that were driving resistance in vitro. And it was particularly interesting to see that we had, in this case, the 5668, we have quite high degree of resistance, over 200-fold resistance, between 220 and 280 times the IC50 of the starting population, so highly resistant. With the degree of cross-resistance to other compounds, we selected mutants against these 3 dihydropyridones here. Depending on the chemical we begin with, the derivative we begin with, we can have lower resistance as well, sometimes around 10-fold. In all of these cases, what we've identified are mutations in a very well-known transporter present on the digestive vacuole of the parasite called PFMDR1. This is the predicted topology of PFMDR1, so this is a multi-transmembrane spanning protein here that's known to be sort of quite pleiotropic drug efflux transporter, which along with PFCRT modulates parasite response to a number of antimalarials in clinical use. And we've been able to pinpoint these mutations, Y290F, and also these mutations, sometimes amplification of this locus, and we know also that these are the exact same mutations we've picked up with some piperazine compounds from another screen. I have to finish. So we now, initially we thought that this would be the actual drug target. We've actually documented drug binding to PFMDR1 using some photoaffinity labeling, but we now think that this is actually quite an effective mechanism of resistance, which probably means that these compounds are actually interfering with heme detoxification in the asexual blood stage. We're not clear about the gametocyte, but now we need to work out the mode of action, which we think will be novel. So with that, I want to thank the individuals that contributed to this work: Sophia Jallet, Ori Lieberman, Sonia Gulati, and Philip Henrich in my lab. From Vicky's group, Leo and Sandra, Steve Hoffman and his colleagues at Scenario, Bob Sinden and Michael Delves from Imperial, Elizabeth Winslow and Victoria from UCSD, and the groups that funded our research: Bill and Melinda Gates, Medicines for Malaria Venture, and also support from NIH. Thank you very much.
We're running late, but there is time for questions.
Berman, Bethesda. Interested in this exciting work. I also interested in your use of methylene blue as the positive control. And I have to say that methylene blue is a clinical agent, even if it turns your urine blue, and has been used for malaria for schizonocidal activity. It's better with another drug, but nevertheless, all the emphasis on a G6 non-hemolytic primaquine derivative to interrupt transmission. Actually, the clinicians in this audience might think of just throwing in methylene blue instead of primaquine and achieving the same results without worrying about G6PD deficiency in the whole issue. I didn't mean to take it away too much for your elegant work, but I just caught the emphasis on methylene blue.
The nice thing about methylene blue is it's much more potent, at least in vitro, than primaquine or tefenequine. Primaquine and tefenequine in vitro essentially don't work. Obviously, metabolites may work, but this is a very potent molecule. It's also very potent against asexuals, which is a property that primaquine does not have. We can't get resistance to it. And so, yeah, I'm a huge advocate of methylene blue, basically.
In the large sense, there's a huge effort to develop a non-hemolytic acetylquinoline to do what primaquine does. And at least for PF transmission blocking, it may already be in the clinic right now.
David, very nice work. I just was wondering, the mutant lines that you made, are they now resistant also in gametocytes?
So that's exactly what we're testing. Last Friday, we were confident enough to call the mutations in PFMDR1 because both groups found the same mutation. So we're now testing whether the gametocytes are cross-resistant. And we're also seeing whether drugs that are known to be acted upon by PFMDR1, such as lumefantrine, mefloquine, amidaquine, will be impacted by these mutations. So those experiments are underway. Okay, thank you.
David, one quick question. I think that what you have shown, I said in my introduction that we have the tendency to focus on stage 5 gametocytes because we know that they are the ones that transmit. But sorry, I said that I said in my introduction that we have the tendency to focus on the stage 5 gametocytes because they are the ones that transmit. But what you have shown is essential. Somehow the dogma according to which early stages would be easily killed by asexual blood stage schizanticides is no longer valid, I guess.
I think that's a very important take-home message. I think our thinking really has been biased by, the long-term use of the 4-menaquinolines that do have activity against the early gametocytes because they degrade hemoglobin at that stage, and also the antifolates. But I think as we look into new classes of antimalarial compounds with novel modes of action, we'll see increasingly that many of those will not have activity against gametocytes, early or late. All right, thank you.
Thank you.